Howdy everyone, my name is Sergio Lozano, and my partners are Cassy, FernandezCassy, Aranda, and today we're going to be presenting on antigen capture, presentation, and recognition in adaptive immunity. So first of all, what is an antigen? An antigen is any substance that can be recognized by the immune system and elicit an immune response. These can be proteins, polysaccharides, lipids, nucleic acids from pathogens, allergens, or even self-molecules in an autoimmune disease. These are the invaders that enter the body and are detected and are acted upon by the immune system. Something interesting to note is that the peanuts and shellfish are considered antigens as they elicit allergic reactions with certain individuals. Within these antigens, there are areas in which the body will react. These are known as epitopes. So what is an epitope? An epitope is the region of an antigen that a T-cell receptor or an antibody binds to. It can be compared to a lock and key, with the epitope being the lock. Paratopes are the key, and the T-cell receptor or antibody molecule that inserts into the epitope. This contact is necessary for the immune system to identify and respond to invading foreign bodies like viruses, bacteria. Essentially, the paratope and epitope are complementary to one another. So how do B and T cells differ in the types of antigens they recognize? The main difference between these two would be that the B cells primarily use antibodies to recognize antigens. This occurs through BCR, in which protein binding occurs to the antigen. On the other hand, T cells primarily rely on antigen-presenting cells, or APCs, to recognize antigens. They do this by binding to the small fragments on APCs. So BCR versus TCR. B cell receptors and T cell receptors are both membrane proteins that are part of the adaptive immune system, with the purpose of recognizing foreign antigens. They have many similarities but can be differentiated by a few qualities. BCRs will primarily recognize antigens in their natural form, while TCRs will bind to the peptide fragments presented by MHC molecules in circulation. The structure also differs with respect to their purpose in that BCRs are composed of two heavy and light chains, each forming a Y shape, while TCRs use alpha and beta chains in a straight chain structure to associate with signaling dimers.
The forms of antigens and the types of macromolecules. Depending on the antigen, various sorts of macromolecules will make them up and have different functions. For example, viruses have nucleic acids for their RNA and proteins for the protein codes. To continue, bacteria have nucleic acids, polysaccharides on their capsules, and lipids. The combinations of these macromolecules form epitopes, which are what allow lymphocytes to be able to recognize these antigens. Types of MHC and their role in the immune system. There are two main types of major histocompatibility complexes, or MHC, MHC class MH1 and MHC class 2. In simple terms, MHC molecules are proteins on the surface of cells that display the proteins from within. It acts as a flag to warn other cells of its status. MHC class 1 is found within all nucleated cells, while MHC class 2 is found primarily in antigen-presenting cells, or APCs. Their main role in the immune system is to present peptide fragments, allowing T cells to recognize infected cells. Antigen processing pathway. The antigen presenting pathway is how the immune system processes and displays antigens to activate T cells. The two main pathways are the endogenous and exogenous pathways. Endogenous pathway is a process that allows the display of antigens within the cell through MHC class 1. The exogenous process involves the process that allows the display of antigens on the surface of the cells through MHC class 2. This system is crucial for detecting infections, eliminating threats, and maintaining immune surveillance. What is the role of dendritic cells in capturing and presenting antigens to T cells? Dendritic cells play the role of capturing antigens in order to take and process them to T cells to elicit an immune response. They will capture antigens from pathogens or damaged cells and transform them into smaller peptides that will migrate into lymph nodes through the lymphatic system and be presented to T cells through peptides on their surface. Once the T cell recognizes the specific antigens, it will activate the appropriate antibodies. Cross-representation. Cross-representation is the unique ability of dendritic cells to present extracellular antigens on MHC class 1 molecules instead of the usual MHC class 2 pathway. This mechanism allows dendritic cells to activate CD8 plus T cells against viruses and tumors even if the APC is not infected.
Where does the development of lymphocytes begin? B lymphocytes and T lymphocytes both develop lymphoid organs, but in different specific areas. B cells mature in the bone marrow, where they start off as stem cells. They mature here until they become mature B cells, which is when they develop immunoglobulins, also known as antibodies. T cells mature in the thymus. However, some T cell precursors are known to migrate from the bone marrow to the thymus, where they mature into fully functional T cells. What types of selections take place during different stages of maturation? There are two main types of selection that occur during the maturation process. These two are primarily positive and negative selections. In immunology, positive and negative selection are two crucial processes that occur during T cell development in the thymus. Positive selection ensures that T cells can recognize self-MHC molecules, which are essential for immune function. If a T cell fails to bind to an MHC, it undergoes apoptosis. Negative selection eliminates T cells that bind too strongly to self-antigens, preventing autoimmunity. This ensures that the immune system can recognize pathogens while avoiding attacks on the body's tissues. Together, these processes create a functional and self-tolerant T cell population. What is the role of negative selection during the maturation process? Negative selection is the process by which the immune system actively searches and weeds out potentially harmful immune cells that could attack the body's tissues. Some immature lymphocytes will bind to self-antigens too strongly. In essence, some lymphocytes will have an affinity to bind and attack the body cells. During the maturation process, adverse selection will be able to detect and trigger a signal leading the cell towards apoptosis. How and why is the vast diversity of receptor structures in the lymphocytes generated? The vast diversity of receptor structures in the lymphocytes is generated through random rearrangement or recombination of the gene segments in the DNA segments coding for the lymphocyte's antigen receptor. These gene segments are split into variable, which is considered V, diversity D, and joining J segments. The large number of different segments plus random combinations leads to their extensive and diverse number. Why have such a large number of receptors is so significant is it allows the immune system to recognize and adapt to any potential pathogen.